It's great to see all your bright and shining faces this afternoon. And as the slide indicates, I'm going to be talking about the McGregor Herbarium and how this has become a resource for understanding botanical questions and floristic patterns across the Plains region. I think a lot of you are aware that about 100 meters behind me in a single-story brick building is the Bridwell Laboratory. And in that laboratory is the R.L. McGregor Herbarium. So it's the actual collection of research specimens that we've accumulated over time here at the University of Kansas. And so you may have that knowledge, but you may not have any other knowledge about how we've accumulated those materials, how we use that information, what significance it is. And so if you've never gone through the herbarium, I encourage you to come over and I'd be happy to show you around. So I want to spend some time giving you kind of a high-level overview of what the herbarium is all about. And then I'm also going to switch gears toward the end and talk about a large project that the herbarium has been involved with for the last 30 years called the Flora of North America project. And so that's what we're going to try to accomplish, and hold your breath. Yay. So before we actually get in and start talking about the herbarium, I want to talk about something that's been referred to as the global collection. And basically what we're referring to here is the 2.5 to 3 billion specimens that have been accumulated in natural history collections around the world. And those form the physical basis for our understanding of a lot of biodiversity across the planet, right? So it's a significant part of our science infrastructure. Unfortunately, a lot of that is black. We can't see it. And the reason is that we haven't digitized a lot of those records. They're not available electronically. The only way you can access many of those data is if you actually visit those collections and extract that information. So we need to do a much better job of increasing that 10 to 15%. I think we got it. Thank you.
Oh, you good?
Appreciate it. Yes. So this is one of the really important things that we're trying to accomplish, is to move the needle and increase the amount of material that's discoverable. So you can think about specimens on the left-hand side as being a primary source of information, right? So we can just take a specimen of Scorzonera from Western Kansas, and that provides us with a couple of things. One, we can have the digital information from the label or annotations, so we can capture that information, and that's a starting point. But we can also take a digital image of that specimen, and those form what are usually called primary extensions. If you then have additional information associated with that specimen, like you may have taken morphological data from it, or you may have tissue samples or field images, those form what have been called the secondary extensions. And imagine if you take lots of specimens and have similar kind of information, you then really start to see the real power of herbarium specimens. That's where things really become important. We can derive species descriptions, evolutionary inferences. We can look at biotic interactions. So there are a lot of things that we can do if we start to aggregate this information from specimens. And this whole concept has become known as the extended specimen network. It goes by a number of other names too, but this is one that you'll see fairly frequently in the literature. So I'm sure all of you have done a little bit of reading and you've bumped into papers that have talked about how specimens are being used to answer questions about biology, ecology, evolutionary biology, those sorts of things. And these are just a couple of examples that illustrate how specimens are being used to address questions that we never imagined that we'd be able to address, right? We tended to think of these in a very traditional way. They give us information about the systematics and the taxonomy of these organisms. But now there are novel uses of these things, and that's where the real power is coming in and the exciting part of the work that we're doing. So how do we implement and sustain this network? Well, there are a bunch of things that need to be done, and they're pretty basic. One, we need to have an idea of what is out there. We don't have an accurate inventory of even all of the collections across the world. The botanists have their act together. They have a pretty good idea of what they have, at least in developed parts of the world, but not so much elsewhere. There needs to be a lot more work in terms of digitization, capturing that information and making it available electronically. Looking at the issues associated with those data. Where are their gaps? Where are their biases? We can't get to that until we really have a lot of that information captured and can start to basically interrogate it. And then working with aggregators. These are organizations that have helped to bring that information together and provide a pipeline so that we can access that information, and then integrating the specimen information with all of the other kinds of data that are out there that we might want to draw in to answer questions in biology and ecology. So botanists have been baling hay for about 300 years. We've got lots of specimens around the world. It's estimated there are about 3,300 herbaria worldwide, approaching 500 million specimens. Here in North America, about 93 million specimens. And only about a quarter of those are currently digitized. This map was put together, I think, about 15 years ago, and it just shows the major herbaria in the United States. And you'll notice that there are some big ones on the West Coast and East Coast, but look at the Central Plains. There's a relative dearth of large collections in this part of the world. And that's why the collection here at the University of Kansas is so important, because it provides a lot of that information that we need to understand floristic patterns in this part of the world. So this is the outside of the Bridwell Laboratory. We were established in 1866 when the university opened its doors. We became a research division within the Biodiversity Institute in 1992. And we're among one of the largest herbaria in the world. We've just cracked the top 100 collections in terms of the size of the collection, the number of specimens that we have. Obviously, we have really good holdings for the Plains region, Midwest, and Rocky Mountains. And what you might not know is that there are a lot of ties to the Biological Survey through time, especially in the last 50 or 60 years, in terms of the collections and personnel. And I'll try to highlight some of those things as we go along. We have a comprehensive collection management policy. This is important in terms of making sure that the material that we have in our care are going to be around for generations of biologists to access. Without these kinds of things that we follow on a daily basis, there's no guarantee that material is going to be there for researchers in the future. We can provide you with a number of services. Some of you have availed yourself of those kinds of services. We do specimen identifications and verifications. So if you have things that you're not sure about, we're happy to work with you to try to put a name on those. We also can help with the preparation and storage of voucher specimens. So if you're involved in a research project where vouchers are necessary, you can come to us. We can talk to you about how to prepare that material in the field. And then we can take over once it's gotten into the herbarium. We will mount the material, digitize it, and make sure that it's available for researchers around the world. So if you have that need, by all means, come and talk to us, and we're happy to assist you with that. Likewise, if you need to access research collections that are in other herbaria around the world, we will help you with that process. Herbaria only loan to recognized herbaria. They don't send material to individuals. So it's important that you connect with us if you need to have research specimens. And we will request those loans, we store the material for you, and then we can provide you with a place to work on that material and then send it back once you're finished with it. And then we also do floristic and vegetation surveys. This is sort of wearing my hat as a member of the Biological Survey. That's where a lot of this work gets done. But we also do it for a variety of other projects. So this is our Rogues Gallery. We've had 7 curators in the collection since it was established in 1866. And we claim Francis Huntington Snow, who looks like he's about 10 years old in this photograph on the bottom left. He's our first curator. He came to the University of Kansas, one of the first 3 faculty members, and he brought a small collection of plants, which we still have in the collection and we still use. And so he's our first curator. The bars just represent the number of specimens from the state of Kansas, and you can see how those have accumulated over time. The one other flag that I want to point out is this one right here, when the Biological Survey, informal, lowercase Biological Survey, was established at the University of Kansas in 1911. The departments of botany and zoology were involved in that. I think there were other ones also, but we actually have some of those specimens in our collection from about 1911 to 1918. There were individuals who were traveling across the state, collecting specimens and depositing them here. So we still have those. I don't know what happened to the zoological collections. I don't know if there are the other collections on campus, but we do have some of the botanical specimens. The other thing I'll mention is you can see this really dramatic increase that occurred from the 1970s to about the 1980s. Ron McGregor, for whom the collection is named, was also the director of the Biological Survey from 1973 to 1983. And I've heard it said by people who worked here during that period of time is he never wanted to see anybody on campus during the week. He expected you to be in the field, baling hay, collecting specimens in streams, doing that sort of work. And so that was the only time he wanted to see you on holidays or on weekends. I think that's probably true based on my knowledge of how Ron interacted with staff. So major holdings in the collection, 2 pie charts. On the left-hand side, you'll see most of the material in the collection are tracheophytes, so flowering plants and ferns. The other big piece of that pie is lichens, and this is a result of a lot of the work that Caleb Morris has been doing with his colleagues in the Northern Plains. So that represents about 10% of the collection, and it's probably the fastest growing part of our collection right now. There are little slivers that relate to other fungi and also to bryophytes. We just have a synoptic collection of macrofungi. Those are mostly from the Kaw Valley Mycological Society. The bryophytes were mostly shipped off. We weren't using those for research, and so there was a large number of specimens that went over to Kansas State University, where there was a bryologist, and those are on indefinite loan there. We also sent a bunch of material to the New York Botanical Garden, where there was somebody doing research on liverworts. So that's what happened to most of those groups of organisms. On the right-hand side are specimen types. Again, most of the specimens are pressed and dried. They're exocati. They're stored in these standard herbarium cases. When you walk in, you see those big gray cases or white cases. That's where most of this material is. But we have about 50,000 packets of lichens that are stored in cases. We have a small spirit collection, so material preserved in alcohol, and then things that we store in boxes. So there is information available about the size of the 9 herbaria that we know to exist in the state of Kansas. That's reflected in the top chart. There are about 840,000 specimens in those collections. 3/4 of the material are at KU and K-State. So here's the KU portion, there's the K-State portion. So that's the lion's share of the material that's accessible here in Kansas. And here you can see the accessioned, databased, and georeferenced material in those collections. So I can't go into a lot of detail about how material was accumulated through time, but I'm going to highlight a few of these things for you. In the last 30 or 40 years, most of the materials come in as a result of floristic work. So studies of specific parts of the landscape like Kansas or other parts of the Great Plains. Work that we've been doing on the systematics or taxonomy of various groups of plants in this region. And then habitat surveys and vegetation assessments. And that's a part where there's a lot of overlap with the biological survey. So oftentimes while I'm out doing work on a vegetation assessment project, say for example, Fort Riley or along some rivers, that results in a lot of voucher specimens that ultimately end up in the collection and has helped to build the collection through time. In the last 15 to 20 years, we spent a lot of time doing digitization and publishing the information, and I kind of want to elaborate on that portion of the activities. We prioritize the material that we're working on by these 3 criteria: geography, taxonomy, and uniqueness. So basically what we're doing is we're looking at things that were collected in Kansas and the Great Plains. We're looking at things that are well represented in the Plains region, and we're looking at things that were collected by KU botanists or Kansas botanists. And those tend to result in a lot of unique records that otherwise are inaccessible in other parts of the world. This is our workflow. So the big part of this is the process of actually capturing that information and making it available to other researchers. So we stage that material, make decisions about what we want to work on. Then we capture the information in databases. We can georeference the material, assign a latitude and longitude if it hasn't already been assigned to that specimen. And then for many projects, we'll go ahead and image the specimen. All throughout that process is data verification. So making sure that the information that we've captured is accurate. And that's often overlooked in a lot of projects. Also, we do a lot of specimen stabilization as a result of the work that we're doing. We see things that are damaged, things that need attention. It needs a fragment packet. It needs to be sewn. So we do a lot of collection management while we're working on these large projects. And eventually we get to a point where we're able to publish that information, ship it out so individuals have access to it. So we've had a number of projects that have been funded that have helped move this whole digitization work along. On the left-hand side was one of the first big ones in 1994. Funding from NSF allowed us to take the information that resided in relational databases that were developed by the Biological Survey and had captured information from the state of Kansas and moved those into Specify, which is software, collection management software developed here at the University of Kansas. And so that was really critical in making sure that we had access to that information that represented about 105 to maybe 110,000 records at the time. We worked on a big eco-forecasting grant, which allowed us to expand the digitization work to states outside of Kansas. So we did a lot of work on the Northern Plains states for that project. And they also worked with Black Hills State University to capture information for specimens from the Northern Plains. More recently, we've worked on 2 TCNs. So these are thematic collection network projects. On the left-hand side is the Tritrophic project. That was really important in terms of about 20 families of flowering plants that were represented in the collection. A lot of imaging took place during the course of working on that project. And then recently we've concluded work on the Torch project, which is a consortium of universities, herbaria, that have holdings from Texas and Oklahoma. And we had about 30,000 specimens that we contributed to that project. We also contribute information to the consortium of lichen herbaria. That's not funded, but it's something that we're able to provide to the community as a resource. So just a couple of highlights on the process of transcribing this information. On the far left-hand side, believe it or not, back in about 1978, they were using mainframe computers at the University of Kansas to do directed collecting. So they would capture information from the collection about a county where something had been collected. So they'd say, Big Blue Stem is collected in Barton County. And they would capture that information for all of the species that they could access, and then they used it to make decisions about what they needed to pick up when they were in the fields. So they were crunching stuff with mainframe computers back then. It wasn't until 1987 that we were able to take information that had been captured, that we were able to capture information in desktop computers and start to make use of that and gradually migrate that over to Specify software. And so we've continued to use Specify for our collection management software since that time. This just shows the impact of some of those projects on the number of specimens that were transcribed since CellGem was in use all the way up to the current time. Georeference is also an important part of the work that we do. We use, we adhere to the community standards in terms of how we georeference the material. Basically, what we do is use the point radius method that's implemented in software that's available on the web called Geolocate. So what this allows us to do is to take a named place, distance, and direction, plug it into the calculator, and it'll give us a centroid, like you see here, and it'll give us an uncertainty radius, which tells us something about the potential fitness of that record for use in research. If you've got a record that has an uncertainty of, say, 200,000 kilometers, that may not be suitable for a lot of purposes in research. But if it's something like this, where you've got maybe 1.5 to 2 kilometers, much more useful for many things that we might be working on. So if you look at the specimens in the collection prior to the 1990s, about 1% of our specimens had geocoordinates on them. So you could find a latitude and longitude, and those were usually assigned by looking at a map, by guess and by gosh. It wasn't until we started to carry around handheld GPSs in the late 1990s that we captured this information, and it became part of the record. We actually put this information on the label data now, or on the labels. We have information about the centroid, we have the uncertainty, we have the datum, and those are routinely captured as part of the work that we do. There was a big effort in 2007 to georeference or retroactive georeference a lot of specimens. And so we added about 150,000 georeference records during that period of time. And right now, about 3/4 of the collection is georeferenced. We do capture images for a lot of the specimens. We currently have about 120 to 130,000 images that are accessible. And these are high quality images. They run about 50 megabytes each. Those are the raw images. We will convert those to a JPEG and post those on the attachment server so that you can actually see those details if you pull up these records. So they're incredibly useful. You can do a lot of identifications or specimen verifications just by doing that. Now, I mentioned data validation is an important part of this process. There's a lot of garbage out there. I don't have to tell you this if you've done any data mining, right? But there are errors that result from intrinsic errors, basically, that are associated with the original label data. And there are things that we call extrinsic error that are resulting from the manipulation of that information. So here's an example. This is about 59 million records pulled from iDigBio. These are for terrestrial plants, right? That's important. Look at all these freckles out here in the oceans. Those are errors. They're either extrinsic or intrinsic errors that are associated with those specimens. And so you can imagine that uncertainty or that error across the entire US. This is a little closer to home. This is a little example that I pulled from our data for Kansas, Cowley County down in the southeast part of the state. If you just look at where the centroids fall outside of the polygon, these are the county boundaries, you'll see that about 1.5% of those records are in error. And those are fairly easy fixes. We can identify what the source of the error is, and then we can make those corrections pretty easily. When we publish this information, we usually ship the material to a number of places. GBIF, or the Global Biodiversity Information Facility, is a really important one. And I know a lot of you make use of that for your research. It's a global information facility. We also publish to iDigBio, which is centered here in the US. And we also will ship material to the Symbiota portals. And so you could go to any of these big data portals and you should be able to access the information that we have shared through those portals. And the really cool thing about GBIF in particular is that we're able to go in and we're able to take a look at specifically what's been downloaded from our collection and used in research across the world. And so those statistics are really important in understanding the utility of the specimens that we're making available to researchers. So here's a snapshot of our stuff that's been georeferenced. This is the entire world, and you can pick out the United States pretty nicely. You can see some more patterns, but we do also have material from South America, Europe, and parts of Eastern Australia. Those are not places where we've concentrated our effort, nor do we have a lot of material from those parts of the world. This zooms in on the US. So you can very quickly pick out, we've done Kansas, right? We've captured all of the information in the collection for Oklahoma and Texas. We've done a fair amount of work for the Northern Plains and the Ozarks. You can see where a lot of the activity has been. But there is a bias here because, like I said, we haven't worked on stuff as much outside of the Plains region as we have within the Plains. So how can we use some of this information to get some basic understanding of what's out there? Well, we can start to look at some of the biases that exist. We can start to identify some of the attributes of the flora. We can look at how the flora is changing, and we can also see how well the collections document the flora. So I'm going to just highlight this with a couple of examples. So here's where I've pulled digitized records for KU and K-State. Those are accessible. The top chart shows the cumulative number of specimens. The orange bars are collections at Kansas State University, and the blue bars are the University of Kansas. There's a big temporal difference, right? So these go back to 1821, which is our oldest record. We have one of those specimens here at KU. But look at what happened just before 1900. A huge amount of work was being done over at Kansas State University to document the flora. Why? Because it was important for understanding the distributions of plants for the economy, for agriculture, for food, for fiber. They wanted to know that. And so there was a lot of work going on during that period of time. And in fact, one of the earliest atlases that was done in the United States was done by researchers at Kansas State University. A.S. Hitchcock published a flora for the state of Kansas in 1899. There was a relatively quiet period from about 1900 up to the end of the Second World War, and that's where the blue really kicks in. That's really Ron McGregor's legacy. Students working with him, as well as a full-time collector, Homer A. Stevens, worked as a full-time collector for more than 10 years. He was a human hay baler. He collected close to 100,000 specimens from every county in every state in the Great Plains. And we have that original material here. So the zenith was in the 1970s. That's when he was out there every place and collecting huge amounts of this. Since that time, since the 70s, we've dropped back into about 4,000 to 5,000 specimens per year. That's what we access. So these are accumulation curves for native and non-native species, again, drawing from information at KU and K-State. If you just look at the black lines, which are the cumulative numbers, this is when a species was first documented in the flora. And you'll notice for native species, there's a really rapid increase, right? Jumps up and then it sort of levels off. And in fact, it becomes asymptotic once you get clear over on the right-hand side. What's it telling us? Well, there was a lot of discovery that happened at K-State early on, and now we're not finding much in terms of native species. Not surprising. This is documenting the obvious. It's a different story for non-native species. A little bit of a jump early on, and look at the black line. It continues to increase. It's telling us that we're continuing to document non-native species in the flora. And you can look at this a little more closely this way. I've pulled in information from Pittsburgh State also. The black line represents the total number of non-native species documented through time since the 1860s. The number averages out to about 3.6 species per year. Every year, adding that many non-native species to the flora. Not all of those things persist. They blink on, they blink off. So if you subtract those non-persisting elements, we're still talking about the blue line here, about 2.7 species per year. That's a lot of species, right? That's close to 550, 560 species that were not here 160 years ago. They've come in since that time. So you say, okay, so what's the impact? Well, we don't know yet for everything, but if we've done a very simple little assessment using invasive index that we apply, and that's what this chart shows you. These bars represent the potential invasiveness of those species that have been assigned to the non-native ones. The stuff on the left-hand side, probably not a problem right now. They look relatively benign right now. The stuff on the right-hand side, those are where our potential problems are, and they certainly are with these in black. So about 2/3 of those species don't look like they're going to be a serious problem, at least based on our understanding of them right now. So the work that I do with the heritage inventory also involves a lot of collecting and documenting rare species in the state. So we know what we have in terms of globally rare species. We can assign state rarity ranks to all of these species and provide that information to organizations. It's used by NatureServe. You can access this information through NatureServe Explorer. And the individual occurrence records that we have, along with zoological records, natural community records, all of that is managed through the Natural Heritage Inventory databases. I've been interested in how well the collections document the flora, and so this is an attempt to try to get an understanding of that. So I just pulled information together for 33 floras that we had a known area and were very well surveyed, and use that to construct a very simple linear regression to look at what can that tell us about the number of species per county across the state. And you get a statistically significant regression. The problem is when you do the calculations and look at the numbers for the counties, it's suspicious. You get a lot of counties in the western part of the state of Kansas that look like they're overinflated, high numbers that we wouldn't predict, and the east is underinflated. So there's something else going on, right? And you probably immediately would think about, well, there's this really strong environmental gradient across the state, right? There's temperature, there's precipitation, there's evapotranspiration. All of those things influence the pattern. So if you introduce a surrogate value for those environmental differences, if you pull in longitude, then you get a much more robust model. And this is an attempt to try to show that. So this is just looking at the documented richness, which we pull from the Biota of North America program. So this is information that this program has pulled from all of the herbaria, from atlases, from published sources. So it represents a maximum value basically for a county. And we can use that as the upper limit. And you compare that to the predicted value, and this is what you get. So basically this zero line, anything above that, the model is predicting those are adequately collected counties. Below that line, at some point, they're undercollected. So why do I believe this? Well, I believe it for a couple of reasons. The red dots. The red dots represent counties in which herbaria, the largest herbaria, are located. Does anybody want to take a wild guess what that one is? Right, Douglas County. That's our collection. You want to guess this one? K-State. Yeah, so that's Riley County. So that suggests to me that, yeah, there's some signal here. And in fact, this has been described as the botanist effect. where you've got botanists, you've got collectors, they go into the collection. So those areas tend to be very well represented in floras. There's additional information. I mean, the other counties do fall out pretty close to the line as well, so they're probably in good shape. These blue dots represent counties in which there are thorough inventories that have been done. I would expect those to fall above the line, and in fact, they do. So what does it look like in terms of the most significantly under-collected counties? They tend to fall out in the western part of the state. We haven't explored this really carefully, but it's probably in a lot of areas that have heavily converted agricultural lands. But we were surprised to see, west central Flint Hills, Ark River lowlands is another area, and the glaciated region up in the north central part of the state. So if I were going to focus on additional surveys, I'd probably focus on those areas. So if you've used any of these big databases or data portals like USDA Plants or the Corps of Engineers National Wetland Plant List or BONAP, you see a lot of similarities in the map. Why? Because those are the original data that they're using are coming from a single source or multiple sources that have been aggregated. They're coming from the herbaria. That's the primary source. Also, a lot of the attribute information that you see in these databases are derived ultimately from the work that collections have put together. So I want to switch gears now and talk a little bit about the Flora North America project. And so I need to add a disclaimer here. This is not truly a continental flora. We've omitted Mexico. Okay, technically that's part of the North American continent. Mexico has a species richness that probably exceeds Canada and the United States combined, right? So we had to draw the line someplace. So we focused on the US and Canada. It also includes Greenland. So this is still a pretty good sized chunk of real estate, about 21.8 million acres and about 15% of the Earth's terrestrial surface. And so we've estimated somewhere between 20 to 21,000 species of flowering plants, ferns, and bryophytes in this part of the world. We don't have a modern comprehensive flora. It doesn't seem like it makes sense, but there were attempts to try to do this, but they never were successful. And the reason for wanting to have a thorough inventory is important across many domains. I don't need to preach to all of you about why this is significant, but it certainly is. And so that provides the impetus for trying to pull this information together. So the idea behind the Flora is to gather all of this information for the first time so that we have all of the names, all of the distributions, the morphological characters in a single source. And the idea was to publish this in 30 volumes over a period of time so that we have this for identification and for a systematic conspectus. So my own involvement in the project goes back to the late 1990s when I got sucked in by this guy, Ron McGregor, who was the curator at the Rocky Mountain Herbarium. Here, Ron is consuming the third large frozen concrete at Ted Drew's on Chippewa in St. Louis. That was a traditional watering hole for all of us when we were out there. And he said, you really need to be involved in the Flora project. And I said, okay. So I got involved in the administration on the board of directors and the executive committee. And during one of the meetings at a local bar over beer and burgers, there were a couple of the individuals talking about the lack of a taxon editor for a couple of families. And I said, I'm kind of interested in knotweeds and their relatives. I'll step in. And that resulted in about a 25-year career as a cat herder. So that's what I've been doing with the Flora project for that period of time. So I'm going to talk briefly about the preparation, the process, and the products. So the initial planning for this dates back to about 1964 when it was announced the first volume of Flora Europaea was going to appear. The North American botanist said, hey, we don't have one. We need to get on the track to get this done. So there were efforts to pull this together in the 1960s and 1970s. That would have been coordinated through the Smithsonian Institution, but it ultimately fell apart because of funding issues. But in the early 1980s is when there was support from Canadian and US professional organizations, as well as funding from the Pew Charitable Trust and the National Science Foundation that got the project off the ground. And the first volumes were published in 1993. Very slow going initially. And in fact, it was so slow that we said, I wasn't involved at the time, but there was a discussion about changing the management style. It was very centralized, top down from the Missouri Botanical Garden. We went to a more distributed management system that allowed a lot more authority to be exercised by the editorial centers across the United States. The other thing that was happening is the loss of that initial funding. And so about 2000, a philanthropic organization, the Chanticleer Foundation, stepped forward and said, we will support your efforts, but you have to produce 2 volumes a year. So that became known as the Chanticleer Imperative. And by God, we got our act together. So we did publish those 2 volumes per year. Since that time, you know, the production level has fallen off a bit. And at the present time, we have 26 volumes out, and there are 4 more in production, with 3 of those containing taxonomic information. So this will include all the native and naturalized taxa. We have partial synonymy. It's not intended to be exhaustive in terms of the synonyms that are applicable to the accepted names. Parallel descriptions, uniform citation of attributes, keys for all of the accepted species. We've got about 19,000 literature records so far, and all of the illustrations in the flora are original. So touch on a couple of the challenges with the project. One of my co-editors always liked this road sign. It's like, yeah, that pretty much described the situation that we're going through right now. So I mentioned this whole management issue that we wrestled with, and seemed to have worked out fairly early in the project. The funding, having major pots of money that we were able to use to support the work. But we have to remember, this is still a volunteer effort largely. You've got hundreds and hundreds of people who are volunteering their time and energies to make this happen. There are relatively few funded positions in the project. Over a 30-year period, you also see a dwindling pool of contributors. People die, people retire. And so how do you get the resources, the individuals with the knowledge to keep this effort going? And then the evolving state of knowledge. One of the big things that's affected us is our understanding of the relationships and circumscriptions of families, going from Cronquist, which is where we started out, to APG, the Angiosperm phylogeny group. All of those things have impacted the project over its life. So talk briefly about process. So this is what we like to think. We've got a very highly industrialized process. It results in a reliable product. We know when the stuff is coming out. And the truth of the matter is, it's more like this. There's a lot of stuff that happens at the local level, and most of that's because people are trying to figure out how to solve a problem. And a lot of times those are really nice solutions, and they find their way up the chain. But that's, I actually think that's one of the strengths of the work that we've done through the association. So like, how do we make sure that we've accounted for all of the taxa in the area, and how do we make sure that they are mutually exclusive concepts? That happens across a lot of different individuals and levels of organization. Bob Kiger, who is our bibliographic editor at the Hunt Institute, likes to sit around and make these pithy pronouncements. And this is one of them they came up with, which is right on the mark. We were talking about how do we transition from a Cronquist classification to an APG classification. And this was his statement that described it perfectly. So how did we implement this? Well, it's been happened in a number of different ways. So one of the early volumes, volume 26, which include the Liliidae, we followed the Cronquist circumscriptions. And if you look at how those genera were treated in the Liliaceae in the broad sense, those genera now map out to about 17 families. You know, we were so far along that we simply couldn't go back to starting point and redo this. By contrast, in volume 17, a much more recent volume, which I was involved with, we had APG4. We had the classification, we had the circumscription of those families, and we were able to actually move that material right in. So it's highly variable across the volumes. For the descriptions, we have controlled vocabulary. We try to make sure that everybody is talking about things in the same way. Parallelism is really important to the project. So an example, if you have 10 species descriptions and you use 5 characters, all 5 of those characters would appear in each of those species descriptions. So they're parallel. Nothing more frustrating than trying to look at a character across a bunch of species descriptions and finding it's missing from most of them. Likewise, you want to make sure that the information that's used in those descriptions appears in the keys, right? you're going through a key and you says something is blue, and that information doesn't appear in the descriptions, there's a problem there. So that's why this has been so important. Also, character extension. So we assume that a character described for a taxon applies to all of its subordinate taxa. All this means is if you say family X has a superior ovary, all of the subordinate taxa, the genera, the species, have a superior ovary unless you state otherwise. And that saves a lot of repetition. And then this is intended to be synoptic. We're not intending for this to be the last word, but certainly an important part of it. So this blob of spaghetti represents the workflow. And all I want to say about it is that it starts over on the left-hand side with authors and taxon editors, and you gradually move things from left to right until it gets over to the Missouri Botanical Garden, where the editorial center is located, the main editorial center, interacting with Oxford University Press, where the volumes are ultimately published. We've had 18 editorial centers. KU served as one of those for 2 of the volumes. There are a number of them that are scattered across the US, but we're winding the project down now, so there are not as many active editorial centers. Authors clearly are the lifeblood of a project like this. We've estimated there'll be about 900 authors contributing to the project once it's done. We've had to do a lot of training and recruitment to make sure that we had the expertise. And in fact, we have had to do a little bit of hiring of a staff botanist to deal with orphan genera, things that didn't have somebody that could work on them. We've also provided small amounts of money to individuals who needed release time from their universities to prepare treatments. And then we provide a lot of resources to the authors. So we've had authors contribute from 28 countries. 13% of the individuals who've contributed to published treatments are dead. They're deceased. So there's a fairly significant turnover that's happened over this the decades of this project. I work with a lot of regional reviewers. So when a manuscript is ready to go out for review, it goes to a regional coordinator. That person is responsible for interacting with individuals in states across their region. So they get it shipped out. They say, here's the treatment. This is when we need the material back. The coordinators then compile that information and ship it off to the taxon editors. So the reason that this is important is because in my case, as a regional coordinator, I can provide a regional perspective. I can pull information from the McGregor Herbarium, our databases, and make sure that it's addressed in those treatments. I mentioned the artwork, it's all original. We've had about a dozen artists who've worked on the project. We can't pay for all of the species to be illustrated. So we do 1 species in every genus in the treatment. We do 1 out of 6 species in the larger genera. And if we have dedicated funding, we can ramp up the numbers. The color frontispiece is something that's really coveted by the artists. They really like to have that position in the volumes. And about 35 to 40% of the funding for the project has gone for just the preparation of the art. If you look across the volumes, there are stylistic differences, but we try to keep those to a minimum within the volumes. Maps almost killed the project. Early on, they were doing these things, these shaded maps, which took forever to put together and to verify. It was just not something that was sustainable. So early on, we switched over to shaded maps, and then there was the realization, hey, this is not an atlas project. There are better ways to get this information. So let's just provide a snapshot of where these things occur based on the information in the flora, and that's what we've gone to. So a few things about products. Obviously, the big products are the published volumes, but you can also go on the FNA website and you're able to access what are called e-floras. They're electronic versions of the floras, but they're not things that are easily searchable. You can find treatments for families or genera or look at species descriptions, but that's about as far as you can go. You also have information for revised since print. So if there are changes, minor changes that have occurred in treatments in some of the early volumes, that's captured and you can track that information on the website. What does allow us to get into more elaborate searches is what's called the semantic search app that's on the website. So you can say, for example, you're interested in all of the plants in Arkansas that have thorns that occur in oak hickory woodlands above 200 meters. You can do that with semantic search because it looks across all of the characters, all of the attributes in those treatments, and will provide that information. So in terms of the secondary products, we've got nomenclature databases, we've got checklists that are coming out as a result of the work, huge numbers of determinations and specimens that are being gathered for the project and other initiatives. This is what the history looks like in terms of the, that primary product. from 2000 or 1993 down to the present. There's the Chanticleer imperative. You can see what impact that had on the project. And this is just a snapshot of one of the posters that was put together that explains the semantic search. So if you're interested in that, I would encourage you to take a look at the FNA website and explore that information. There are two big databases. I don't know how many of you use Tropicos or IPNI. Tropicos is maintained at the Missouri Botanical Gardens. IPNI is at the Kew Gardens. This is the primary place where most of us who are working in with vascular plants will go for information about nomenclature. And so a lot of information, the vetting of names, verifying that information is fed into these large databases. If you used any of these things, you're probably benefiting from the work that's happened with the flora. Certainly the taxonomic backbone that's produced is something that's mostly derived from FNA with some exceptions. And also some of the attribute information is being pulled in by these other data portals. So just to wrap stuff up, this was the love of my life for about 10 years. There are no more beautiful plants than penstemons. A lot of stuff in the Western US. I could talk to you for hours about this stuff, but I'm not going to. All I want to do is just to mention some of those derivatives. In addition to the treatment that was produced for the flora, which ultimately covered 239 species, we looked at over 500 names and verified them. Over 100 types that were undiscovered in collections were pulled out and determined. I looked at about 30,000 specimens in 30 herbaria. So those determinations are in collections around the United States, and also made a lot of collections myself that were used in the project. And the cool thing is that my emphasis was on just getting this treatment written, but all of these other things are now contributing to other research projects. There are about half a dozen research projects that are going on, making use of the tissues, the specimens, the character matrix, all of those kinds of things. And I think that's probably the most satisfying part of this whole effort. So I want to conclude with this. This is the mantra that's been adopted in recent years by the association. Done is better than perfect. Now there's a tagline here too, and that is that you really need to have some idea of what your standards are and which one of those standards are non-negotiable, which ones you're not willing to change, because there are certain things that you don't want to give up on. And that really has to do with the veracity information that's going into these products, right? So the accuracy of the data, that's something that we don't compromise on. But there are a lot of other places where you can compromise and move the project along. So I think this is a really good mantra to follow. So that's the last slide that I have. Some acknowledgements, but I'd be more than happy to answer questions if you've got them now. So thanks very much. Helen.
I have a problem.
Yeah, those are both good questions. The first one was, if we tried to do this project again, starting it now, would we have the expertise that we would need to produce the flora? And the second one, is there training that's occurring to try to fill those gaps that appear to be developing? In answer to the first question, I still feel that way about it. I think that, that 13% turnover, it's probably closer to 15 to 20% now. We're losing a generation of individuals, of botanists who are trained to do this sort of stuff. And so this may be our last best chance to do that. Now, there are efforts to try to do more of that work that would feed into this, but there are really different interests that I think exist in a lot of academic institutions. And so that's part of the reason that I think it's so important for us to certainly make sure that we do the best we can with the information that we've got now, so it's available for other researchers. And the other thing that I've seen happen in recent decades is a lot of the expertise now resides with paraprofessionals and with amateurs who are really not amateurs. They're really quite well versed. And that's certainly playing a major role in other parts of the world, less so in the United States. But I think you see it in a lot of other countries where that kind of resource is really going to be critical. Tropics, a huge amount of discovered biodiversity. How are we going to ever get to a point where we understand that? And that's where we're going to really need to be drawing in a lot of individuals who maybe not trained at the same level, but still have the capacity to do that work and contribute. Yeah.
You mentioned this effort stopped at the Mexican border.
Yeah.
Is there some parallel effort to do this for Mexico or Mexico and Central America, or if not, what are the major sources of Floristic information for that. purpose?
Yeah, so most of the work that I know about that's happening in Mexico, there was a question about, the Flora project stopping at the border, Mexican border. So what's going on in Mexico and Central South America to do similar sorts of things? There are some big floristic projects that are going on in the New World, in Central America. Most of those things, the big ones that are nearing completion or have been completed, have been directed by herbaria, oftentimes in Europe or in the United States. So if you go look at Central American countries, for example, a lot of that work has been done through Missouri Botanical Garden or New York Botanical Garden, or by botanists who are working out of the large herbaria in Europe. So there's still a lot of this, I guess you could say, colonial influence that happens in some of those initiatives. That's why I think there's more emphasis in a lot of these countries to try to build that infrastructure and expertise so that they can do that work themselves. Mexico has a really robust system to capture the information. As far as I know, there's not a single concerted effort to try to produce a flora like this. And you could argue that books are antiquated, right?
Yeah.
There are better ways to present this information, and we hear this all the time. I still love to hold a book, and I think a lot of other people do too. But that's one of the things that we're trying to figure out right now. How do we make sure that the flora continues after we wrap up that last volume? How do we make sure that it's a living document? And that's where some of the real challenges are. Yeah, Gene. So there was a question about this kind of conspicuous break in terms of the number of digitized records in the Northern Plains. That's mostly a bias that exists in the data because we haven't worked as much outside of the Plains region. So I expect that you'll start to see a lot of that information fill in as we work on those additional specimens in our collection. There are a lot of them there. We just haven't worked on them yet. We have about probably 100,000 specimens have yet to be captured. Yeah, so question about updating some of the earlier volumes of the flora. Basically, what's happening right now is we're making minor changes to those as we capture that information. That's through the page that's maintained on the FNA website. So you can see where some of those minor adjustments. But in terms of how do you rearrange the furniture, like what's happened with Amaranthaceae, Chenopodiaceae in volume 4, they're all one big mass now, right? And there are many other similar changes that have occurred. Those we don't have a method for dealing with efficiently. So everybody's sitting around talking about ways that they might be able to accomplish that. So right now there's no formal plan to, publish a new volume that would address those sorts of issues. You're kind of stuck with what we had from that period of time, and then using crosswalks to try to relate the information to more current classifications. Yeah, Jerry.
This week, of course, shifting politics is very much on the minds. And I'm wondering the broad spectrum of what's been accomplished and what still needs to be accomplished. Do you find an undertone or overtone of political politics influencing what you think you're going to be able to accomplish? I mean, views of climate change are shifting, and I won't get into all the political things that are shifting. But are you vulnerable to that? Do you see vulnerabilities?
So the question is about the vulnerability of the work that we're doing to political whims, I guess. And certainly there's always that risk, right? But I think the important thing is that as we do this work, we demonstrate the importance of and the value of the information that we're providing as it relates to societal needs. I don't want to think about this thing in strictly utilitarian ways, but I think that's absolutely important that we show how the science that we do is important to anybody on the street. That's one thing that we need to make sure we do. I have not seen a lot of that the kind of winds of change through the work that we've done on the Flora North America project over the 30-year history. There may have been stuff early on that I'm unaware of, but by and large, we were pretty much removed from that because of the sources of funding that we were using. NSF certainly provided a little bit of money up front. Subsequently, it was mostly coming from philanthropic organizations and places like that. So in some respects, we were sort of divorced from that. I don't know of any situations in which we've had complaints or issues raised by, let's just say by politicians about the information that we're generating. But I do know that in some states that occasionally does happen with information that's gathered by heritage inventories, for example, that's coming off of public or private land. We've seen examples where somebody gets worked up about information that's been gathered and shared. And so it's actually affected the availability of information in some states. So I don't know if that really answers the question very well for you, but
It does.
yeah, it's
And I don't know if there is-
it's a tough one for sure. Yeah. Anything else?
One more quick.
Yeah.
Where were the original herbarium collections on campus? Were they in Dyke Hall or?
Most of them were up in Snow Hall.
Snow Hall?
Yeah, and in 1965, they moved out to West Campus. That's when the first part of the building was constructed, and then 1985 is when the addition was added. So yeah, they may have been someplace else. They probably were in one other building on campus that I'm unaware of, but Snow Hall is where most of that stuff was housed. All right, thanks very much.